Oh yeah, absolutely. If your doctor tells you that you need to take medications, then you should take medication.
Yeah, because if they don't follow their diet, I'd rather have them on medicine than have a heart attack.
Yeah, that's true. Okay, so that's true. Lesser of the evil, I guess. Anyway, so I said once again, there's a lot of things you can do to reduce your cholesterol before you resort to medication. Okay, so that's the consensus in the whole world anyway. Okay, except in America. In America, your cholesterol slightly high. Okay, yeah, here's Lipitor. Okay, so that's you go take Lipitor. Okay. Anyway, so this is the cholesterol level. It's always interesting. You can actually go watch these videos at home if you want to learn more about all the details of the different types of fats. Okay, so we will talk about amino acids right now. So amino acids, the proteins, they make up all the important molecules. All the structural components, all the enzymes are made up of proteins. And then amino acids are different from carbohydrates and fats because amino acids actually has nitrogen. N, okay? So carbohydrates and fats are only carbon, hydrogen, and oxygen. And but amino acid has nitrogen in it. Okay, so nitrogen is an important element. All right, so now amino acid they all have this structure, carbon in the middle called the alpha carbon. Okay, one hydrogen group, and then the left side here is called the amino group, and then the right side is called a carboxylic acid group, C11OOH. Amino group is NH2. And then in this position here, you put an R group. R refers to the side chain of the amino acid. Right, so there are actually 20 different types of amino acid. Okay, they all have the same basic structure. They all have the same NH3, C11OH, and H. But it's what you put in the side chain that makes different types of amino acids. Simple ones like these. Okay, non-polar ones you put in HCH3. Okay, so these are the non-polar ones, and then you can actually put in positively charged ones. You can see here, lysine, arginine, histidine. There's actually a positive charge. These are negatively charged ones, aspartate, glutamate. These actually contain an extra negative charge. These are hydrophobic ones called aromatic, non-polar aromatic ones. You can see here with a benzene ring here, like phenylalanine, tryptophan, tyrosine. And then you have some polar ones, which contains additional OH groups. Okay, so OH groups and sulfate groups here. Okay, so sulfur group. Cysteine contains sulfur, and you can see proline here got an extra charge here, and then you have asparagine and then glutamine. Okay, which got an extra C11O group. Okay, it's got these polar side group. So the properties of the amino acid actually differ substantially. Okay, so it's what you put in the side chain. Okay, so you can put in a non-polar group, and then you can put in some positively charged group, negative charged group, and then a big, hydrophobic groups like these benzene ring groups, and then you got polar groups. Okay, so right, so this is the differences between all of these amino acids. Okay, so when I studied biochemistry in college, I have to learn the structure of all of these 20 amino acids. Okay, so you guys don't need to do that. I'm not going to ask you to. Remember, I had a teacher come in, and when I was studying biochemistry in college, she just come in and say, draw cysteine. Okay, so draw cysteine. Okay, remember the side group. Okay, so now you don't have to remember all the structure, but you should know that the amino acid property actually differ substantially depending on what kind of group they are. Okay, so if it is a non-polar one versus like hydrophobic and hydrophilic ones. Right, so all of these are different types of amino acids. So amino acids also polymerize together to form bigger proteins. Okay, so the way that they combine together is also called dehydration synthesis. You can see an OH and an H here. All right, combined, and then H2O leaves the molecule, just like what we talked about yesterday in both the carbohydrates and fats. And H2O leaves the molecule, forming the peptide bond. Okay, C11ON. And then this gives you can keep joining amino acid like that. Okay, so you can keep adding amino acids to make your protein. Okay, so right, so now there is actually you can also do that. There's actually a directionality in building up a protein molecule. Okay, so it turns out that your body will only put amino acid to a free carboxylic acid terminal N. Okay, so your body will only put amino acid, we only put more amino acid from the C from the carboxylic acid end. Okay, so right, so here it would actually not put any amino acid on this end. The body will only put new amino acid to the free carboxylic acid terminal. Okay, so the HEF and OHA reacts, H2O leaves the molecule. So you always have a number one amino acid which has a free NH2 group called the N-terminus. You only add a new amino acid to the carboxylic acid group here. Alright, so at the very last amino acid, you will have a free carboxylic N called a C-terminus. Okay, so there's directionality in the protein. So every protein will begin with the free amino acid NH2 group. Okay, so you only add new amino acid to the C-terminus. Okay, you do not add any new amino acid to the N-terminus. Okay, so that's how your body makes proteins. Okay, so there's no reason why you can't chemically put amino acid on this end, but it's just how your body makes protein, that there is a direction. Right, so you need to know these terms, because these terms are very common when people describe proteins. The N-terminus of the protein versus the C-terminus of the protein. Okay, so the N-terminus is the beginning of the protein, and then the C-terminus is the end of the protein. Okay, so this is how any biochemist or any biologist would describe proteins. Alright, so now you can actually string them together. Actually, this is how. So the primary structure is the type and number of order of amino acid sequence that goes into the chain. So you do add one amino acid at a time, and you lengthen the chain. Okay, so you in a mature protein there could be a small protein can have 60, 70 amino acid. A big protein can have 300-400 amino acids. Okay, you rarely see proteins bigger than 400 amino acids. They don't fold properly. Okay, so if there are more than 400 amino acids going into a single protein, so you can. But there's definitely an order. Okay, so the number one is always the one with the N-terminus, and then you keep adding you keep adding into the C-terminus, and then and eventually when it stops, okay, then the protein you can start folding the protein. So the order in which the amino acid you go in is called a primary structure. Right. So once you have synthesized the protein, all right, once you have put the amino acid in correct order, the protein will actually start folding. Right, so the common secondary structure that a protein will fold, these are called beta sheets and alpha helix. Okay, so occurs when a sequence of amino acids are linked together by hydrogen bonds. Okay, there's a vague definition for that, but what you can think of is it will start folding among its neighbors first. Okay, it will start folding, interacting with its neighboring amino acids first, folding into either beta sheets or alpha helix. Some other structures. Okay, so two common secondary structure are these beta sheets and alpha helix. Right, so you can think of just folding among its neighboring amino acids first, and then the whole protein would come folding together. Okay, so and then when the whole thing comes folding together is called a tertiary structure. So of course, when certain attractions are present between alpha helix and beta sheets. Okay, it's a vague definition, but what you can think of is when the whole protein folds together now, okay, so after folding with its neighbor, the whole thing now can fold into a bigger, overall three-dimensional shape of the protein. It's called a tertiary structure. And then now finally, when you have more than one chain of amino acids that are, sometimes a protein can be made up of several chains that are separately folded, okay, then the protein has a quaternary structure. Okay, when there are multiple chains that are synthesized independently, but then when they come together to build an even bigger protein, that's called the quaternary structure. Okay, so I put a little bit of note here. The tertiary structure, known as the overall three-dimensional shape of a single chain of polypeptide. Right, so quaternary structure is formed when more than one polypeptide chain forms a large multi-unit protein. Okay, so I do want to play this video because the video kind of explains it a little bit better how the folding works. Okay, so see if that. Let me pause it. Okay, because the mic is not on here. Okay. Start again.
You could probably name some.
Sorry. This isn't. Oh, it's 100% already. It's not loud enough. Oh well, we have to. This is all the way up here too. It's not very loud. Oh well, let's just play anyway.
You hear about proteins a lot in the media today. You could probably name some high-protein foods right now if I asked you to. Foods like meat, beans, and nuts are often the first to come to mind. What are proteins? What are they made of? And why are they so important? First of all, proteins are made of one or more chains of amino acids. You can visualize a protein like a beaded bracelet, where the beads are the amino acids, and the overall bracelet is the protein. The sequence of amino acids is what gives the protein its shape and allows it to do its job. Proteins have a huge variety of jobs in the human body. Do you remember the hormone insulin? Insulin's a protein. Most enzymes are made of protein. Your muscles are made of protein. Even your bones and teeth are made largely of protein. That's why proteins tend to get so much attention, and they're very important to our health. Let's look a bit closer at the amino acids that make up proteins. First of all, there are 20 different amino acids that we humans need to make the proteins in our body. About half of those amino acids are essential, and about half are non-essential. You'll remember that essential means we have to eat them or drink them, because the human body cannot make them, or cannot make enough of them to fulfill our needs. Here are the names of the 20 amino acids: histidine, isoleucine, leucine, glycine, methionine, phenylalanine, threonine, tryptophan, valine, alanine, aspartic acid, asparagine, glutamic acid, serine, arginine, cysteine, glutamine, glycine, proline, and tyrosine. Do any of those sound familiar? All 20 of those amino acids are made of the same 4 basic parts: a central carbon, an amino group, which contains nitrogen, an acid group, and an R group, or side chain. The only part that differs from one amino acid to another is that R group. The rest is identical. In this example, you are looking at the amino acid alanine. See how the side chain is a CH3? In another amino acid, everything would look the same except that side chain would be different.
I thought it was a video on. This is the one that I actually. This is the one that I. Yeah, this is actually too long.
This familiar gelatin dessert.
Yeah, don't have to worry about that one. All right, so yeah, so this is the how proteins work. Okay, now proteins actually make up everything. Okay, so it makes up your enzymes, antibodies, everything. Okay, so practically all your muscles are made up of proteins. Now the video does mention essential versus non-essential amino acids. Okay, so that's why sometimes you will hear people talk about high-grade proteins versus low-grade proteins. Okay, so high-grade proteins are the ones that are rich in the essential amino acids that we need. Okay, typically those are animal proteins. Okay, such as you know, milk, and when you eat meat. Soybean too does contain good essential amino acids, but there are actually some low-grade protein, things like gluten. Okay, so that is very common in wheat. Wheat actually has a lot of protein in it. Pasta also has a lot of protein in it. They contain a lot of gluten, but gluten is not considered a high-grade protein. Okay, even though it's very high in protein content, it's actually not the kinds of amino acids that we like to consume. Okay, so so there's also the difference between high-grade versus low-grade proteins. All right, so now this is the yeah. So proteins actually makes up everything. Okay, so when the all of your body's enzymes, everything is needed. So the term denatured means when protein structure is disrupted. All right, so this happens when you heat it. Okay, it's usually when you heat it, folded protein, a folded protein like that, requires a stable temperature. So when you heat it, the chain actually unfolds, and then they would just then randomly pile on top of each other. Okay, that's what denaturation means. Okay, so when you heat it, you usually will break open the chains, and then they then just coil on top of each other and forming like a mesh, just like when you boil an egg. Okay, so when you put an egg or when you fry an egg, okay, the liquid portion they turn solid. Even when you cool it, then they stay permanently solid afterwards. Okay, this is denaturation. Okay, once it's done, it's actually permanent. Okay, so you cannot go back. Okay, the function of the protein will be permanently lost once you heat it, and that's also the reason why you die when you're exposed to high heat. Okay, or living organisms die, or bacteria or organisms die when you boil them. Okay, because the proteins are denatured and all the functions are lost. Right, so that's called denaturation. This actually is a little too long. Actually, you can watch it on. So we'll move on to DNA right now. So which is an essential part of the molecule. Now, we'll talk more about this in chapter 8. So this is just a brief introduction on DNA.
Structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate.
Alright, so the 3 and 5 prime actually is determined by the position of the carbon. Okay, so if you study chemistry, you learned it in chemistry, but you always name the carbon. This is carbon number 1, right next to the oxygen ring. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5 is the one that is sticking out. Okay, so that's why DNA has this 5' and 3' name. Okay, the 5 and 3 actually comes from the position of the carbon. Okay, so don't ask me why is that the case. The chemists have determined that they want to name the carbons according to that position. Okay, so it's just by convention that whenever you have a ring-shaped molecule, the one on the right next to the oxygen is considered carbon number 1. Okay, the carbon, so this carbon number 1, 2, this is carbon number 3, carbon number 4, carbon number 5. 5 is what's sticking out. So that's why DNA, the sugar molecule in the DNA, has orientation. This is called a 5' end, and that's called a 3' end. Okay, so it refers to the position of the carbon. Okay, so.
Free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not